Hello and welcome to this video tutorial about using lab protocols and actions in Sesame. For this tutorial, we will use the Scheherazade module. This video is about actions in lab protocols and using them to manage projects, record and track activities, item statuses, etc. The other parts of lab protocols are covered in separate videos. Let's start by opening one of our lab protocols. Go to the main window in Scheherazade and go to Options in the menu bar. Under the Lab Resources submenu, click on Lab Protocols. The Lab Protocols window now opens in the board. I'll call up all of this lab's protocols. Under the Search menu, choose Search by Owner and leave asterisks in the fields here. Now, all of my lab's protocols appear. Let's open Protocol 8834. Basic fields in protocol records were covered in a separate video. Next, we will show how to add actions to a protocol. Actions are a set of attributes that may be added to an item when a particular lab protocol is selected, and they may track activities, outcome, statuses, etc. of the item that they are added to. Actions are defined in the Actions Lab resource. Let's open that now. In the main window, go to Options, scroll down to Lab Resources, and then choose Actions. The Actions window now opens in the board. To see all the actions in the lab, I will do a search by owner, leaving an asterisk in all the fields. The top part of the window shows the results of the search. When I select an action from the list, the details are shown in the lower part of the window. Managing and creating actions is discussed in a separate video. As you can see, my lab has numerous actions listed here. Your lab may also define as many actions as you want to track. Actions may either represent binary outcomes or a range of outcomes. Actions may be used for different kinds of reporting, data mining, like what protocol is better on some protein types, or what is the distribution of outcomes, or how long does it take on average to do something so that a pipeline may be set up correctly, etc. Let's search again this time. I will restrict my search to look for actions with membrane protein in their name. I'll enter MEMB asterisk into the action field here. Now let's add two actions to the protocol that I have selected. This is quite easy. Just select the action from the list in the top part of the Actions window and drag and drop it into the title bar here at the top of the protocol window. You can see that the action is now listed and attached here. Let's add another action. I'll drag and drop that again now. Let's save these changes and choose Save Lab Protocol under Protocol in the menu bar. To see the details of an action, I may open any of these actions quickly in the board by highlighting the line I want to open and choosing the blue I button to the right. The action opens in the board. Actions may be added to almost every Sesame item type. Now I will show how to add actions to a protein record. Using a search previously, I have retrieved some of my lab's proteins here. Let's open protein 99. To be able to add actions to this protein, first, a protocol must be selected, and then the actions from the selected protocol, and only those actions, may be added to the protein. Scroll down to the Actions field here. This protein already has some actions attached, as you can see. Another lab member, Leah Kerr, has added some. Any member of the lab may add actions to Sesame items. Let's add another action. Select the green button to the right of the field to add an action. A pop-up window appears with a drop-down list of the actions from the selected protocol. Let's add an action. I'll start with purification complete. When an action is added, it is automatically added to the database record. We do not have to save this manually. And when an action is added, the user who added the action, the time, and the protocol number is also recorded here. An action may be deleted, but only the last action that was added and only by the creator of the action. For example, let's say I forgot to add an action, polishing concentration stop, before the action I just added, which was purification complete. Just press the red minus button to delete the last action. Now I will try again to delete, but I cannot because the last action does not belong to me but to Leah Kerr. Now I'll fully correct this record by adding polishing concentration stop and then purification complete. Records may be searched by the actions added to them. Let me demonstrate in protein records. In the main page, under search in the top menu bar, scroll to protein. Under the side menu, choose search by action. In the search window, all actions that are connected to proteins in your lab are auto-populated. If we wish, we may choose a protocol from the drop-down menu item Action Lab Protocol here. If we choose a protocol, only actions associated with it will be shown. I will still leave this blank. You may designate multiple choices here, choosing present or not present as your search requires. Let's search for form cell growth. Now all protein records that have this action added are listed in our search results. Click on one to open it in the board. I will scroll down to the Actions field, and we can see that the action Form Cell Growth is listed here. Let's move to look at the reports that we may create for any item that has attached actions. In the last window under Reports, let's look at Protein and choose Last Actions. We may choose to look at the last one or last two actions. Let's choose 2. We may also restrict the report by lab protocol, time period, etc., but let's leave these fields blank. Now, a report opens in the board listing the last 2 actions for proteins in our lab. If we want to see all the actions added to one or more proteins, we may use the Actions by List report for proteins. The particular protein's database numbers may be entered here as a list of numbers separated by space, comma, semicolon, tab, or a new line. Alternatively, a named list of database numbers, in this case protein database numbers, may be created using the DBNO list item or database number list item. Then, a list of proteins will be selectable from the drop-down list here. I will choose June proteins from the drop-down list. This report will list the actions in the selected time period by default in the last week. I will leave the remaining boxes here as they are to call up all actions added to these selected proteins. The report elbow grease under proteins shows how much effort was put into the proteins in the lab. You can do it without selecting a particular protocol, or you may select one if you are interested in seeing how much work was put into the proteins under a particular protocol. The report may also be restricted by time period or particular action. If no action is selected, it will show all actions. Let's keep this blank to show all actions. Now, a summary of actions appears, showing protein number, ORF number, and an action count for reporting needs. The report action totals will show, for a given time period, all the action counts that may be grouped by multiple levels and then lab protocols. Let's group this time by action. Now, we may compare different protocols with this report, etc. There are other detailed videos covering the use of protocols in Sesame. These videos cover the basics of creating a protocol onto more advanced features. Please visit our website for these videos and for additional videos, help pages, and more information about Sesame.